So a couple of questions have come up about the online quizzes. On occasion there might be a typo. On occasion there could be something completely wrong. So what I want to encourage you guys to do, and I've talked with a few of you about this in my office hours, is I want you to practice articulating your argument. Okay? And it doesn't have to be like, you know, Macbeth or, you know, like Shakespeare prose or anything. I just mean writing complete sentences. Okay? If you send it to me from your phone, take the time to capitalize and punctuate. Okay? You is Y-O-U. Okay? This is professional conduct. And we become really relaxed with technology. And I want to make sure that I'm encouraging my students to behave in a professional way so that you guys are better trained to be professionals when you get out in the real world. Okay, so that's the reason for it. I'm not asking you to do anything that I don't stick to on my own. And if you want evidence of that, come see me. And I will show you. I have examples of manuscripts that I've submitted, and I think there are some reviewers that really just don't like me. I've had grants where I think they hate me, and they write really hurtful statements. And sometimes you get a chance to respond, and you can't respond like, what a jerk. Right? You have to be like, an interesting point. Okay? Which is professional code word for take a hike and, you know, go jump off a cliff. But you just say it in a different way, and I want you guys to practice that. So if you're hearing from me on an email of, I'm not really understanding your argument, or what is the actual question, it's not me trying to blow you off. It's me trying to encourage you to take it to the next level and defend your perspective. If you want to tell me about where it is in the book, or if there's a website, insert it and quote it. Put it in quotations. And don't just list the web link and make me read through like 30 pages of a website to see possibly what it is that you're trying to prove. Okay? If you find a primary paper, wow, it would be really cool if you had the PDF and you just add it there. You can access PDFs for most of these journals, these scientific journals, for free as a student, right through Klein Library. Okay, if you don't know how to do that, ask Kelsey or I will show you how to do that. I can probably email you instructions. You can go to the library, they can easily tell you. Okay, these are all things that are, for example, you disagree about a diagnosis, or one nurse to another, you don't agree about, you know, someone wants to give pure water in the saline bag, on the IV bag, and the other one says, I think we should use this, you know, almost pure water of 0.9%. It's, you know, almost the same thing, right? You're probably going to need to put a paper there to explain why you possibly might kill the patient by putting water in their IV. Okay? And there's all sorts of colorful words you could use, but you won't use those. You'll use professional tone. Right? So that's what this activity is. I encourage it. I want you to do it. There were a couple of typos that some of you pointed out. Thank you for that. It makes the class better when you guys can point them out. But it also is super nice when you get to practice articulating a disagreement or an argument with essentially someone who's your supervisor. Okay? And then we have a professional relationship in that way. Cool? All right, so let's dive into arguably the most important part of today, which is, what did the tree say to the wind? It's not inflammation, just so you're wondering. Not inflammation. Some of you are like, inflammation! Neutrophil! We got like a twitch. Should I repeat the question now that you're distracted? What did the tree say to the wind? Leave me alone. Nicely done. What does a lost sock look for? What does a lost sock look for? Its soulmate. All right, so if you look at our syllabus, we're right on track. Should have completed the pre-quiz 1, and by the end of the week, or pre-quiz 2, excuse me, by the end of the week, post-quiz 2 is due. I didn't send out an announcement this time because I told you I wouldn't. I want to try to take the training wheels off and have you guys do things on your own. But you know, if you have any questions about the flow, it's going to kind of be the same system every week. A pre-quiz, the lecture, and then the post-quiz. That's kind of how it's supposed to work. These are supposed to be like bite-sized pieces. This is sort of the main course. That's why we sit here for 2 hours, right? Getting really full on information. So today, we're going to launch into a rather critical lecture as it relates to the semester, because so many of the diseases that we're going to study from this point on are going to have an element of inflammation associated with them. And the first statement I want to make that you should write down is, inflammation is a good thing. Inflammation is a good thing. Now, wait a second, I thought you just said it was part of a lot of disease states. Well, yeah, that's inflammation that's out of control. Another name for that would be chronic inflammation. So in patients that have disease states where inflammation is out of check, it would be considered chronic inflammation. And it has a lot of the same attributes as acute inflammation. Acute inflammation is a critical part of normal wound healing. In fact, if you don't have a phase of acute inflammation in wound healing, you probably will not have wound resolution. You will not heal the wound. Okay, so it is needed. But just like all things, you can have too much of it. And when it sticks around long term, otherwise known as chronic, is where we see all these problems. So just flashing forward to some of these diseases. We'll see at the end of the semester atherosclerosis. There's chronic inflammation associated with atherosclerotic lesions. Rheumatoid arthritis. There's chronic inflammation associated with these lesions that typically target the smaller synovial joints first, and then they make their way to bigger ones. Not always, but many times. Okay? We'll see disease states in the kidneys. We'll see disease states in the liver. We'll see evidence of chronic inflammation, and we'll also see evidence of a wound healing process where there's a leftover remnant, like a scar. And that's fibrosis. And we're going to talk a little bit about that today. So we're going to set up much of the semester in today's lecture in 2 parts. Take the first part, and then we'll take a break. Okay? I'll weave in a little bit of my research because it's related to inflammation, because I deal a lot with normal wound healing. And I also deal a lot with biomedical implants in my lab. And anytime you put something in the body as a foreign object, there's a response directed towards that material. And there's a wound healing response. And there's a perturbation where it alters the normal wound healing process. And we can measure that. We can look to see what happened. Did this biomaterial cause an exaggerated inflammatory response, or was it quote-unquote normal? Okay? So we'll look at a lot of things today in the category of inflammation so that we can set the stage for the rest of the semester. But I said in the very beginning, hopefully you caught that inflammation in of itself is a good thing. In fact, it's critical to the survival of organisms. We as an organism need to be able to eliminate threats or invaders, foreign invaders. We need to be able to handle infectious pathogens. We need to be able to repair damaged tissues. And I told you that inflammation correctly, normal inflammation or acute inflammation, is part of the normal wound healing process. So it helps to repair the tissue that's damaged. Inflammation is an answer that the body provides as a protective mechanism, and it's defined by many immunologists as part of this innate immunity that we have. So I'm introducing a couple of concepts here. What does innate immunity mean to you? What does that phrase mean to you? You're born with it. Who said that? Nice. You're born. What else?
You don't need prior exposure.
What's that? You don't need prior exposure. Agreed. I like both of those. What else?
It's found in everybody.
It's found in everybody. Okay? Even in some really crazy genetic disorders we'll talk about, there's still an inflammatory process. Okay. What else? Anything else? Is it customizable? Or is it pretty much the same every time? It's pretty much the same every time. It's pretty much the same in all of us. So if you have an inflammatory episode, we would use probably some of the same pharmaceutical strategies from one patient to another patient called anti-inflammatories. We'll talk about some of those. Steroidals are very aggressive anti-inflammatories. Okay? COX inhibitors are huge anti-inflammatories. And they're very effective in all of us as long as we don't have an allergy to them. We'll talk about allergies in a couple of weeks. Okay? And this innate immunity that you're born with means that it's not acquired. You don't have to learn it. It's in everybody. It's the same every time. Okay? You're born with it. All of these would fall into the category of being part of this innate immune system. So our innate immune system is really called our first line of defense. But inflammation happens to be sort of a fence rider, if you will. It kind of sits between the first level of defense and the second level of defense. Okay, so let me explain this slide, and then we'll move to the next one, and I'll take your question, Troy. So the first line of defense here, we've got our innate immunity, where we look at things like epithelial barriers, like our skin, our mucous membranes. These are all things that we're born with that are not acquired, that help protect us against microbes and harmful agents. They could be not just physical, but they could be mechanical as well, like hairs, okay, cilia. They could be, like the respiratory cilia that move in the trachea and the respiratory bronchi. They actually physically have this escalator that takes the mucus retrograde to get rid of the debris and the dust that comes in as you inhale. Okay, so that's a great example of a mechanical. Is it physical? It is physical, but it has a mechanical characteristic because it's actually moving and doing something there. Then we have biochemical, like our enzymes. We have a lot of enzymes in our mouth, a lot of enzymes in our tears. So these tears have these enzymatic activities that help protect us against some of the harmful agents that come in contact with that mucous membrane. Of course, one of the worst things you can do, but it feels so good, is like rub your eye, right? You guys ever do that at the end of the day? You just get both of them going, like, roll, right? It's like a dog that scratches that right part of their ear, and they're like moaning, right? Are you a moaner when you like rub your eyes? Sometimes it feels really good, but I like to wash my hands first, and then I'll just go at it, okay? But it's really not a good idea, because that's putting in a lot of possible bacteria. So our physical barriers, innate immunity, and then in a couple of weeks we're going to look at this adaptive immune response, where we're going to start seeing some very cool things that happen that are learned. They're programmed. You have to be exposed to them. There's a process that takes place before it gets going. Okay? So we're kind of dividing up this segment into innate, first line of defense, and we'll call second line of defense is inflammation, which is our second slide. This is part of our innate immunity, but we kind of call it our second line of defense. Our third line of defense, which is the next slide, then I'm going to come back to this, is our specific immunity. Our specific immunity. We're not going to talk much about it today. We're going to come back to this slide in a couple weeks when we talk about the adaptive or the specific immune response. This is where we get a lot of autoimmune disorders. This is where we get a lot of vaccinations that show up. This is where we have the ability for antibodies or antibiotics to work. Okay? So the learned portion or the specific portion we will come back to. I want to back up one slide and sit on the second line of defense. Before I do that, there's a question in the back.
I was just wondering, do you know of any extreme differences between the inflammatory responses
Across species. So in the mammalian kingdom, it's very similar. It's very, very similar amongst all mammals. Now, what happens if you cross over to birds or reptiles or things? I don't know the answer to that. I definitely know more about the human, but we use a lot of model systems to replicate the human. in different types of disease states, and they're all mammals. And that's because we have all the same cells that are present. What happens when you cross over out of the mammalian kingdom? I don't know the answer to that. It would be interesting. I wonder how much is conserved. It's an interesting question to ask, but I just don't know the answer. You've reached the limit of my knowledge. Mammalian kingdom is it. All right, second line of defense. Back to that. Any other questions? Okay. So second line of defense. This is a busy slide, but it's a great summary slide. So let's park here for a minute and let's describe some of the things that are going on. Okay. This is a transverse slice or a longitudinal slice through a blood vessel. So you're seeing the inside of the blood vessel and you're seeing the flow go from the left side to the right side of the screen. And then you see in 202, we characterize these flat monolayer cells, which are called what? Endothelial cells. And what do the endothelial cells do? They protect. They what? They resist abrasion. What else do they do? Anything else? They do secrete certain things. They do absorb certain things.
Control permeability.
They control what?
Permeability.
They control permeability. That's where we're headed. What else do they do? There's a long list. We could actually be here for a while. But there's one other thing that's critical that I want to get to before. I wanted two things. Permeability was one. All those other ones are correct.
They make it frictionless.
What's that?
They make the frictionless.
Frictionless. Yeah, someone already kind of said that. What's their luminal interface? What are they in contact with?
Cells.
In the blood. Thank you. Wow. Okay, that was hard. We'll go slower. Don't worry. So in the lumen, this is 202. In the lumen, there's blood flow, and the endothelial cell is exposed to the blood surface. It also is exposed to the basement membrane and can allow things to actually cross. So it controls permeability. And it also has a very important anticoagulative characteristic. It actually keeps the blood from clotting off. It's called an antithrombogenic surface. You with me? This is important because if you lose that antithrombogenic surface, you actually have a site for cardiovascular disease to begin. And we'll talk later in the semester about what causes damage to the endothelium. But there's all sorts of different things, like high blood pressure, like smoking. A lot of the toxins in cigarette smoke are actually endothelial damaging agents. Okay? Too much lipid in the blood. Right? It's a very viscous interface, so it's more aggressive if you have you have hyperlipidemia where you have lots of fat in the bloodstream. That's actually a more aggressive fluid than a sort of less fatty blood flowing through. Okay? So it controls coagulation and it regulates the permeability of the vessel, allowing things to come across. So now you can see there's other things in the blood, like platelets, have an obvious coagulation role. We have our circulating monocytes, or circulating lymphocytes. We have plasma proteins that actually help with the coiled osmotic pressure to bring back fluid into the bloodstream from the tissue, right? Because water follows solutes. So the plasma proteins that stay in, like albumin, encourage reabsorption of fluid out of the extracellular space. And then we also have these polymorphonuclear leukocytes. So we have the ability where between the endothelium we have these tight junctions. They're connected. And when they're connected like this, there's actually a solid surface and it's not very permeable. Well, we can actually lose the connection, create a gap, and let stuff come through, or what we call extravasate out of the blood vessel. Okay? When we do that, we create a gap in a space, fluid can leak out, some proteins can leak out, and cells could actually migrate out. They don't leak, they actually physically have to like crawl through. And we call that fancy word diapedesis. Okay? So we'll talk about all these things, but this slide really helps to characterize it. So why would we want to make the vessel leaky and let stuff get out? Well, let's pretend down here in the extracellular space, extracellular matrix, we actually have microbes, right? We have an infection. Well, you want these circulating cells, specifically you want the monocytes to get out and become, see how they change their architecture? When they leave the circulation and they extravasate out, or they go out into the space, they become what we call activated tissue macrophages. And those activated tissue macrophages are going to go phagocytose and gobble up the bacteria. And that's a critical part of an inflammatory process, whether it's acute or whether it's chronic. Now, if it's acute, those macrophages come on the scene and then they eventually go away relatively soon. Chronic means that they would stick around. We'll talk about that later. So the blood vessel that circulates throughout the entire body, like you saw in 202, is carrying rich blood or valuable blood, nutrition, and it actually has the ability to deliver certain cell types to actually stimulate inflammation. Okay? So where does the inflammatory response come from? It comes from the vasculature. Right? If you have a tissue that doesn't have a vascular supply, you cannot generate an inflammatory response. Make sense? If you can't generate an inflammatory response, you can't get fluid out delivering nutrition. You can't get monocytes out making macrophages and cleaning up the debris. And I told you that inflammation is a critical part of normal wound healing. So if you have a non-vascularized tissue and it becomes damaged, theoretically, does it heal? No. Or maybe it's very poorly vascularized, so does it heal well? No. What are some examples? Just to prove my point. What? Tendons are, they don't have a high vascular supply. Ligaments as well. There's another example in that kind of family that's a better example that's relatively considered to be avascular. No blood vessels. And if you've ever had damage to this type of tissue, you know it doesn't heal. You usually have to have surgery. Cartilage. How many of you have actually had a meniscal tear, or you've torn an ACL and there's cartilage damage, right? Did you pop a couple of ibuprofen and in like 3 weeks it was healed? No, that cartilage does not heal well because cartilage is, for the most part, non-vascular. Okay? So evidence that inflammation is important for a tissue to be able to heal. Because you need a blood supply to get all these important nutritional agents out and to get all these important cells out to actually clean up the debris, to clean up the mess. So we're going to come back to a lot of these things, but the endothelium regulate whether these things can move out. And there's going to be cytokines that we'll talk about later in the lecture, mechanistically, that have influences on whether the blood vessel shrinks in diameter, which we call vasoconstriction, or whether it dilates or gets bigger, which we call vasodilation. If we dilate and get bigger, what happens from one endothelial cell to another as you dilate? They get stretched out, and eventually there's a space. Can you visualize that? You go from this diameter tube to that diameter tube relatively quickly, and the endothelium can't adapt. What happens to their tight junctions? They actually separate. So now you've created a gap. So an agent, like a histamine, which causes aggressive vasodilation, allows for big gaps to happen for fluid to leak out. Okay? Histamine's a cytokine that's readily available during an inflammatory response. For example, how many of you have allergies? What do you take to combat your allergies during allergy season? An antihistamine. You block the release of histamine so that you don't vasodilate that tissue that's being responsive. Maybe it's your nose or your lungs, okay? Or maybe it's a food allergy, or maybe it's a skin allergy, and that histamine response causes an inflammatory response. Redness, right? Swelling. We'll get to all of these here in a second. Okay, so summary slide. gives us the concept that the vasculature is critically important to being able to harbor an inflammatory response. Second point, inflammatory response is critically important for wound healing to take place. For example, cartilage, avascular, doesn't have any blood vessels, doesn't heal very well. Okay? Let's look at some of our clinical sequelae, or our clinical results. That's a word that means kind of, what's the clinical consequence? Well, the characteristics of inflammation are fourfold. We have what we call the 4 cardinal signs of inflammation. We have dolor, or pain. We have rubor, what we call redness. We have calor, which is heat. And then we have tumor, which is swelling. So why do we have all those things? Well, if the blood vessel is vasodilating and fluid is leaking out, you get more blood flow into the tissue. That's where the swelling happens. The blood that flows into the tissue is warm because it comes from your circulation. That's where the heat comes from, right? It causes redness because there's an increase in perfusion, right? If you get embarrassed, you become flush in the face, right? And that's increasing the perfusion of blood to your dermis extremity as a sort of a reflex. So that causes the redness. Now the pain, where does the pain come from? Well, pain is triggered by stimulating nociceptors, which are the pain receptors, and they are sensitive to the increased pressure in that tissue that's putting pressure onto the nociceptor, causing the pain. Or it could be pain because the nociceptor is in tissue that's actually been traumatized. Okay, so if it's an inflammation because of a tendon tear or a ligament tear, then you've actually damaged tissue, and you probably have severed or torn nociceptors of their pathways. So let's talk about acute inflammation and this rapid response that takes place. We have 2 main things. We have vascular changes and we have cellular changes. So if you look up top we've got our normal situation. This is a capillary bed. We've got an arteriole coming in on the left and a venule leaving on the right. You can see outside of the bloodstream the occasional lymphocyte or resident macrophage that are kind of considered what we call sentinel cells. They hang out in the periphery kind of looking for any action so that you can respond a little bit faster if there was a foreign invader or a harmful bacterial agent. So the important thing is the relative numbers. I mean, what you can see here is we have a few, but we don't have that many compared to the bottom. In the bottom situation, we have inflamed tissue, right? And the first thing that I want you to be able to appreciate is this vascular change. We dilate the arteriole aggressively. We dilate the capillary bed, and we dilate the venule aggressively. And what that does is it increases blood flow. You make the tube larger. Blood flow goes up into that, ankle because you twisted your ankle, right? And you know after you twist your ankle, in the next couple of hours it swells up. Okay? So we have these vascular changes, and we know, we can appreciate from that previous diagram that when we dilate those vessels, we increase the gaps between the endothelium, and what we say is we've increased the permeability. So by vasodilating, you increase the permeability in a mechanical way, and you allow more blood flow into the region, and you allow a more leaky vasculature into the tissue. Well, that's a good thing if you want to heal, because if you leak fluid into the tissue, you're bringing oxygenated fluid in. You're getting an opportunity for cells to migrate out. They're going to clean up the mess. Okay? You also have vasodilated the venule side, so you can increase the uptake of metabolic byproducts. So you've increased the delivery of nutrition, and you've increased the removal of waste products. That's all going to facilitate healing. So those are our vascular changes. And the cellular changes, notice, look at the number of neutrophils that actually migrate out of the vasculature. And next, after the neutrophils, the second responding cell, because the neutrophils are the first responding cell, what's the second responding cell? They just take a little longer to get into the tissue. Macrophages. So very classically in inflammation, you have a first wave of neutrophils that come on the scene, and then you follow it with a second infantry of soldiers, and those are called macrophages. Okay? And what I'm showing you with these hand gestures is a graph that comes up and then it comes back down. And I'll show you a graph here in a slide or two of where these peaks actually show up relative to each other. If we continue to look at the vascular changes and the cellular changes, the vascular contributions are vasodilation. Now, vasodilation accomplishes a couple of things. Number one is you get an increase in blood flow to the region, and you leak fluid out into the tissue, allowing for stasis of that fluid, residency of that fluid to remain in the tissue. Okay, so you're getting that nutrition out instead of bringing more blood flow in and making it leave the region right away. By actually having it leaky, it remains, or it stays, or it remains static, where you have stasis of that fluid in the damaged tissue until it can be used up, and then you remove the waste products on the venule side. You increase the permeability. If you increase the permeability, again, it leaks out, creates a stasis of nutritional fluid in the damaged tissue, which is where you want it. That causes an increase in onychotic pressure in the extracellular fluid. That's what that stands for, ECF. An increase in onychotic pressure in the extracellular fluid means that you have what? More what in the extracellular tissue? What's that? More stuff, otherwise known as like plasma proteins. Okay? So more stuff like plasma proteins are in the extracellular matrix. So what does water want to do? It follows. So it leaks out. So you've promoted the leaking of nutritional fluid out of the vessel into the tissue. That's why when you have inflammation, it has to swell. That's a good sign, actually, to an extent. You want to encourage some level of inflammation in a wound so that it actually can heal. And that's contrary to kind of a lot of our thinking is every time you have inflammation, we should take an anti-inflammatory. Well, maybe not. If it's chronic in nature, that's a different story. If it's acute, it may be part of the normal wound healing response. You may not want to subside it completely because you may actually delay wound healing. So if you increase the vasodilation, the cells pull apart, the endothelial cells pull apart, fluid is leaking out, and proteins are leaking out. We call these two transudates and exudates. So let's characterize what the difference is between a transudate and an exudate. You can see up top, we've got sort of a normal vessel under homeostasis. So we've got plasma proteins that are mostly inside the lumen of the vessel. We have hydrostatic pressure. This would be synonymous with blood pressure, right? The blood's pressurized by the heart, so there is a hydrostatic pressure head that wants to encourage the fluid under pressure to go from high to low. So it wants to leave. Does that make sense? It wants to go from inside to out. Well, then we have this arrow coming the opposite direction back into the bloodstream. So this one is suggesting that the fluid is leaving via hydrostatic pressure, also known as blood pressure. This arrow is suggesting that fluid wants to come back into the lumen of the vessel. And the driving force here is this colloid osmotic pressure. That's set up by our plasma proteins, primarily by albumin. Albumin is a very large plasma protein, and it is what stays in the bloodstream and encourages fluid to come back in. Now, in a transudate situation, you have fluid only leaking out because the gaps in between the endothelium are not large enough for these plasma proteins to come out. And you can see that the pressure vector of hydrostatic pressure is greater, and so this leaks more fluid out, and we're returning less of a force back. And so the fluid leakage in the extracellular space contributes to the swelling. Well, in a more aggressive vasodilation, we actually get large enough gaps that we get fluid and protein leaking out, and we would call that an exudate versus a transudate. So the things that cause vascular changes or modifications, and I've alluded to histamine already. I said it was a vasodilator, potent vasodilator. It's released by mast cells. It's released by other cell types as well, but mast cells tend to be a really, really large primary source of histamine. Histamine increases the capillary permeability. And the other compounds that are in this same family are shown here. We've got kinins. The kinins, another name for them, also known as, are called cytokines. Some specific examples that cause an increase in capillary permeability are a kinin family, a cytokine, known as TNF-alpha. We call it TNF-alpha. It stands for tumor necrosis factor alpha. Why is it named that? Well, because scientists name things weird. Okay? We do. So it was a cancer investigator, and we'll talk about this a little bit more in cancer biology, but in our unit on cancer bio. But this tumor necrosis factor alpha was extracted out of tumors, and it was discovered as being a potent angiogenic factor, meaning it stimulated new blood vessel development. In order for blood vessels to grow, they actually take advantage of an increase in capillary permeability. That's how they grow, because they grow from a sprout. So if you have an existing blood vessel, and you want to grow another blood vessel over here to this tissue, you actually make a break in the vessel, you increase the permeability, and you sprout out a branch to form, from vessel 1, you form vessel 2. And what causes this breakage to happen in the endothelial cells to kind of migrate down and then line the inside of this new vessel are things like tumor necrosis factor alpha. Okay? So this is a strategy that tumors use in order to survive. They grow new blood vessels to keep their growing mass perfused. Okay? So now it's called tumor necrosis factor alpha, although in our lecture today it has nothing to do with tumors. Okay? But it's a factor that's well used in a lot of places in the body. Another one's called interleukin-1. I don't know the backstory on interleukin-1, but it's another cytokine that causes an increase in capillary permeability. And then the third one is this compound known as VEGF. VEGF actually used to have a name in the early days known as vascular permeability factor. That was its old name. I like that name better because it relates to this lecture. So the early name of VEGF was vascular permeability factor, or known as VPF. So if you look in the literature, like in the 1950s and 60s, you'll see it talk about VPF. And then an individual named Judah Folkman kind of popularized VEGF. He was a cancer biologist. He was an MD, Ph.D., so he was in the clinic as well as in a research lab. And he was very interested about how to stop cancer growth and development. And he kind of coined, with some of his colleagues, vascular endothelial growth factor, VEGF. Vascular endothelial growth factor. Well, what VEGF has is an activity much like tumor necrosis factor alpha, is it causes an increase in the permeability of the existing blood vessel so that a sprout of endothelium can migrate out and cause a new blood vessel to grow. So this is such an important process. There's not just one factor that does it. There's probably 2 to 3 dozen that are described in the literature that have this type of activity of increasing capillary permeability. But I'm only going to have you know essentially like 4 or 5 of them. Okay? Deal? But these are kind of big ones that you're going to run into in the literature. When you see VEGF or you see TNF-alpha, or you see an interleukin family, hopefully the exposure in a general pathology class will be like, I don't know exactly what it does, but I do remember learning about that, and I know that it has something to do with vascular permeability, I bet. Okay, and then you'll move on and learn a little bit more and you start connecting more dots. Okay? So these are the agents that can cause these aggressive vascular changes. And those vascular changes happen first, and then the cellular changes follow almost immediately. Almost immediately. You know, there might be a little bit of a gap, but almost immediately. So the exudate is the end game, where you get leakage of plasma and proteins, and then the cells can come out right after that. So if we take this background that we've been talking about and we start applying it to acute inflammation, we can look at a couple of different steps. And I've got a video. We're going to watch a portion of the video, and then you can go to the link later and watch the rest if you'd like. But I think it really helps to put into motion what's happening. We have steps to follow in order to encourage cells to get out. So I've just talked to you about how fluid gets out, and hopefully I've convinced you why getting fluid out in an inflammatory response is so important. Well, fluid alone isn't going to do it. In fact, you want the fluid first, because you want to put nutrition into the tissue, but you then want to bring in the appropriate cell types in order to do the activity. How do we get the cells there? We have to recruit them, number one. Well, then we have to turn them on. We have to activate them so that they know to do something. You don't want to circulate in the bloodstream macrophages in an active, aggressive state, ready to just sort of clean up anything they see. So they circulate in an inactivated state known as a circulating monocyte. And then when they get out of the bloodstream, they get activated and turned on to go do what they're supposed to do. Okay, they go through puberty really fast. Okay? Then you want to have them target where they're supposed to go. That's a fancy word known as chemotaxis. So chemotaxis is movement towards a chemical gradient. So if you have an inflammatory event in the tissue, that damaged tissue may be releasing all sorts of different signals. These signals could be things like vasodilators. These signals could be macrophage stimulators to cause a macrophage to come towards it. Okay? These signals are targeting cells that are nearby in order to get them out of the blood vessel. So the idea is, if they're circulating here, they actually need to migrate towards the edge, right? That's sort of like getting near the jump zone. Then you want them to attach, but first they kind of slightly stick, and as they slightly stick and come unstuck, they start rolling. So it's called rolling and tethering, because they stick and get tethered, they get unstuck and they roll. Eventually they adhere. And once they adhere in a spot, they kind of lay down. And then when they lay down, they look for a little gap. It's like, all right, I'm going over here and I'm going to squeeze right through that hole. Or they go up, I'm going to squeeze this way. Or, you know, it doesn't matter, it's three-dimensional. My goal is to extravasate, to get out of the bloodstream, to move towards, chemotax towards this signal that's telling me this is where you need to be. Okay? It's really elegant how this works. So what I want to do is go to this website. I should have preloaded this, sorry about that. See that rolling? Hang on. Yeah. Do you see how that... How come my sound is... One second.
You turn your master volume all the way down.
Oh yeah, I did, because I was like rocking out and I needed to We're having user error problems here. There it is. Thank you. That's more better. Let's try that again. That was just a teaser. Inner life of the cell. A diary account.
While red blood cells
Whoa,
are
that's
carried
on
away
now.
at high velocity by a strong blood flow, leukocytes roll slowly on endothelial cells. P-selectins on endothelial cells interact with PSGL1, a glycoprotein on leukocyte microvilli. Leukocytes, pushed by the blood flow, adhere and roll on endothelial cells because existing interactions are broken, while new ones are formed. These interactions are possible because the extended extracellular domains of both proteins emerge from the extracellular matrix, which covers the surface of both cell types. The outer leaflet of the lipid bilayer is enriched in sphingolipids and phosphatidylcholine. Sphingolipid-rich rafts raised above the rest of the leaflet recruit specific membrane proteins. Raft's rigidity is caused by the tight packing of cholesterol molecules against the straight sphingolipids hydrocarbon chains. Outside the rafts, kinks in unsaturated hydrocarbon chains and lower cholesterol concentration result in increased fluidity. At sites of inflammation, secreted chemokines bound to heparin sulfate proteoglycan on endothelial cells are presented to leukocyte 7 transmembrane receptors. The binding stimulates leukocytes and triggers an intracellular cascade of signaling reactions.
Okay, let's pause for a second. So in the very beginning of the video, you see this leukocyte kind of rolling along the endothelial surface, right? Then they zoomed in and they showed you these selectins. These selectins have a component on the endothelium and on the leukocyte, and they try to interact. And as they interact, they're a weak bond, so they break. Why do they break? Because there's that pressure head behind the cell because it's sitting in blood flow, right? So that selectin isn't a strong attachment, and so what happens is it rolls. You see how it was rolling across the surface. So it rolls, eventually it's going to find a spot, because as those selectins are being signaled, it's triggering the endothelial cell inside the cell to say, okay, something's cruising along our surface, triggering us to activate. Maybe we should actually grab one of these guys and let them move through. So as they upregulate their expression of integrins, that is another molecule that will have a stronger attachment, and that's what actually anchors that endothelial cell down. Okay? I'm going to fast forward to 643. So get some popcorn and watch 144 to 643. But I'm going to go right to here, because this is what's relevant for us right now.
Chemokines secreted by endothelial cells bind to the extracellular domains of G-protein-coupled membrane receptors. This binding causes a conformational change in the cytosolic portion of the receptor, and the consequent activation of a subunit of the G-protein. The activation of the G-protein subunit triggers a cascade of protein activation, which in turn leads to the activation and clustering of integrins inside lipid rafts.
Okay?
A dramatic conformational change occurs in the extracellular domain of the activated integrins. This now allows for their interaction with ICAM proteins displayed at the surface of the endothelial cells. These strong interactions immobilize the rolling leukocyte at the site of inflammation.
See how it
Additional
stops?
signaling events cause a profound reorganization of the cytoskeleton, resulting in the spreading of one edge of the leukocyte.
Here it's
The
going to slip
leading
in
edge of the
between.
leukocyte inserts itself between endothelial cells, and the leukocyte migrates through the blood vessel wall into the inflamed tissue. Rolling, activation, adhesion, and transendothelial migration are the 4 steps of a process called leukocyte extravasation.
Okay. That help? Is that helpful? Just to kind of see it in motion? pretty crazy. I mean, that animation it's like a sci-fi, doesn't it? Seem like a sci-fi movie of some sort? I mean, it's a popcorn, 5 minutes, you get all the other, G protein activation cascades. It's pretty exciting. Okay, you might have seen some of that in other classes, like in 205. Okay, but this is where the outside of the cell is triggering the inside of the cell to do something. First, the selectins are being triggered. Then you trigger integrins, right? And the whole goal is to adhere the circulating white blood cell so that it can actually diapedese, transmigrate across the wall of the blood vessel out to the extracellular tissue.
Is that rolling process, is that always happening, or is that only taking place when the leukocytes are beginning to be recruited?
It's a great question. The question is, does the rolling happen like right now? Is like things just rolling right now? Usually not. It's usually in a sort of a pre-activated state where you're going to start activating the rolling of those white blood cells.
Okay.
Okay.
And then once it gets like to the spot, that's when the
The integrins are anchoring. We're going to talk about it right here, right? So the selectins. So first up, margination. I'm going to add a couple of steps. They said 4. I'm going to add a couple more. Okay. It's not quite that simplistic. It's a really good video, but more because like the animation's like stellar. Okay, and I could never recreate something like that, so I just have to borrow other people's. But it's really good. It's showing what's happening. So margination. First, to answer your question, if we want to stimulate the rolling to happen, first we need to have the white blood cells move to the margin. So instead of flowing down the middle of the bloodstream, they need to move to the edges. And in order to do that, one way that we facilitate that is we actually stimulate a controlled coagulation. So if you clot the blood at a relatively gentle level, you'll make it more viscous. And making it more viscous is going to push the cellular elements to the outside and allow the fluid to flow down the middle. Okay? So it's not like you shut the whole bloodstream off. You just start activating certain parts of the blood to coagulate it and marginate. Move the cells to the edges with a controlled coagulation. Now you've got the cell on the surface and the selectins can see their adhesions and they start rolling. That rolling event of triggering those selectins every time they break intracellularly signals the upregulation of adherents, or integrins, and these integrins are responsible for the adhesion. So the rolling and tethering is the selectins, the adhesion is the integrins, and the transmigration is down here when you see the macrophage, the monocyte starting to move out, becoming a tissue macrophage. This is transmigration, otherwise known as diapedesis, as it moves through. Okay? And then the last step is chemotaxis, which the video didn't even talk about. The chemotaxis is moving down a chemical gradient to get where you want to be in the tissue. Okay, so we've got 5, 6 steps depending upon how you count. The video only talked about 4, but I think there's more like 5 or 6 that I want you to be aware of sequentially. Activation of the monocyte happens as soon as the selectins are triggered. As they start to roll, they become activated.
So it says up on here that the selectins are on the reticular integrins on the one side on the top left.
Right.
There's also integrins on the other.
Well, so the selectin molecule, right, is on the endothelial cell, and it binds to this antigen, right, which is the Silyl Lewis X modified glycoprotein. That's what it binds to. That's what the video was describing. So the selectin is on and expressed the endothelial cell. Okay, but its binding site that it binds to is on the leukocyte. Now, if you look at the integrin is actually expressed where? On the circulating white blood cell. But it's going to bind and react to its integrin ligand, which is called the ICAMs. You see the video talking about the ICAMs? So the selectin is on the endothelium, and its binding is on the white blood cell. And it's flipped in the integrin molecule. The integrin is on the white blood cell, and its ICAM, right, its binding ligand is actually on the endothelium. It's a good question.
So is the integrin ligand, is that only on a tissue that has inflammation, or is it on all the cells?
The integrin ligand gets upregulated. See how it's not expressed here? It gets upregulated by the endothelium when it starts to roll. And the integrin itself moves from a low affinity state, you see how it's got this little tent cap,
Right.
to a high affinity state when the white blood cell starts to roll. So both the endothelium and the white blood cell actually get activated because of the rolling.
But how does it know which tissue to go into? Because that's what I was asking.
How does it know which tissue to go into?
Yeah.
So these get activated only where these things are actually rolling. And so the way that they roll and become activated is because of the signals that the tissue creates from the damage. So the chemical agents in the tissue go up here, like histamine gets released, and now all of a sudden the vessel gets permeable. So damaged tissue down here will release histamine. And that will cause the blood vessel to dilate. And now it becomes leaky. Do you see what I mean?
Yeah.
If it becomes leaky, you actually start to stimulate coagulation. If you stimulate coagulation, you start to move marginate, you start to move these white blood cells to the edge, and this whole thing happens. Other questions? This is pretty important, so I'm trying to show it many times, but in different formats. Okay? Those are good questions. Any other questions? Okay, so if we look at after we've activated, what next? Well, we've got the macrophage in the tissue, and the end goal is going to be phagocytosis. We want to engulf either a bacteria or debris and clean up the bacteria that's present or clean up the damaged tissue or damaged cells that are present. So the end goal of the macrophage is actually like a cleanup mode. But in order to get there, let's take the example of a microbe. We utilize these receptors called toll-like receptors that actually help to identify the microbe and bind to specific recognition portions of that microbe. And then it signals it through a process known as opsonization, which means that it's getting it ready to be phagocytosed or engulfed. Then it engulfs it, brings it in, kind of invaginates the whole thing, surrounds it with a membrane, right here. Okay? And then this merges with a lysosome, a vesicle, that actually has digestive enzymes. And now you have a phagolysosome, and these digestive enzymes can start chewing up the bacteria, or if this was just cellular debris, it would happen in a very similar way, and this cellular debris that was maybe necrotic tissue would start being digested because of the enzymes. Okay? So you have receptors identifying the tissue or the microbe. You have opsonization signaling it for phagocytosis. Phagocytosis engulfment, kind of the same thing. And then you start digestion by merging it into a phagolysosome, and you stimulate degradation of the microbes using your enzymatic digestion. Second thing that happens, okay? Second thing that happens is you actually utilize this phagocyte oxidase in the membrane of the phagolysosome to generate, in the presence of oxygen, free radicals, reactive oxygen species. Now, last time we were together, I told you that these reactive oxygen species can cause lots of tissue damage. Remember? Remember I showed you the Fenton reaction? So, this is a situation where does the macrophage get oxygen for this process in the tissue? Remember, oxygen can easily translate through a phospholipid bilayer, right? It's a gas, permeable. So where does it come from? Vasodilation allows for nutritional fluid to flow in that's highly oxygenated, right? That's why you need that vasodilation to happen first. That gives you your source of oxygen to run through this oxidase system to generate a reactive oxygen species. Okay, fine, but why is that a good thing? Well, this reactive oxygen species can destroy the microbes. They're harmful to bacteria. So now you have a vesicle that has a bacteria inside of it, and you're cranking out harmful reactive oxygen species to kill the microbe. You're sanitizing it from the inside of the cell. It's pretty darn cool. As long as that process is happening under control and the macrophages are on top of it, then the microbe levels go down, right? And the infection goes away. What happens if the bacteria outpaces the level of the macrophage activity? In fact, if the macrophage actually loses and the bacteria wins, and it ruptures the macrophage, what leaks out into the tissue? ROSs, right? Causing more tissue trauma. So that's why if you have an inflammatory event, an infection, it might be a teeter-totter of managing the infection, but if the infection gets out of control, right, and starts to pus and those kinds of things, then you have the bacteria is lysing your macrophages, and that's causing additional tissue trauma because all of the harmful agents that were there designed to kill the bacteria are leaking out into the surrounding tissue. So it can actually progressively get worse. Okay, that would be an infected wound. So this process of creating these reactive oxygen species is known as this respiratory burst. So this respiratory burst is not just enough that our macrophages are equipped to digest away tissue and debris with enzymes. You also create this respiratory burst using arginine as a precursor through iNOS system to generate nitric oxide, and this phagocyte oxidase in the presence of oxygen to generate reactive oxygen species. Those create these free radicals of oxygen known as this respiratory burst, and it's great for killing bacteria, but it could be less desirable if the bacteria wins and you lyse the macrophage. Make sense? Okay. So, the leukocytes, how do they damage tissue? Sometimes we'll see in chronic infections or chronic inflammation that white blood cells that I'm characterizing as the good guys are actually the bad guys. Well, how can they cause damage? They can cause damage if the reactive oxygen species levels get too high because the bacteria wins. The other thing is if they stick around too long, then they start creating this fibrosis. So if you look at the tissue injury on the left, these are all the different things that are inside of a macrophage, right? We have enzymes like proteases. We have chemotactic factors. We have coagulation factors. We have nitric oxide. We have toxic oxygen metabolites like reactive oxygen species. So if you lyse this macrophage because you lose the battle, you've dumped out reactive oxygen species, enzymes, chemotactic factors, coagulation factors. This is going to chew up the tissue. The other thing that's inside is vesicles, because this macrophage makes all these different growth factors. They make growth factors like platelet-derived growth factor, PDGF, fibroblast growth factor, FGF, transferrin growth factor beta, TGF-beta. They make a bunch of different cytokines that stimulate fibrosis. Fibrosis is a wound response to try to patch up damaged tissue with excessive amounts of collagen. And that's what you see when you have a scar, like on your arm. You fell and you cut yourself really aggressively, and what grew back was tissue, but there's a big scar. That's fibrotic scarring. Okay? That's your body's sort of last resort to try to patch up the hole so there's not this big gap in tissue. Well, a lot of these factors like fibroblast growth factor, TGF-beta, platelet-derived growth factor, all of these stimulate remodeling of collagen. That's that word of collagenesis. Collagenesis is new collagen, and you start to remodel it. You form a scar. So if these guys stick around long term, you can develop tissue injury and you can develop tissue scarring. And we're going to see tissue injury and tissue scarring in cardiac infarcts. You're going to see them in strokes in the brain. You're going to see them in cirrhotic liver. You're going to see them in renal infarcts in the kidneys. Right? You're going to see tissue injury and you're going to see fibrosis in all sorts of diseases throughout the semester because inflammation is at the root. Okay? We're going to take a break here in a couple slides. Some evidence of where inflammation is strong, or how specialized it is, or how, in other words, how cool it looks. Okay? This is a macrophage. It's a moving leukocyte, but it's outside the body, so we call it a macrophage. And you can see this filopodium in the upper left. And so it's kind of crawling from the lower right to the upper left, and it's kind of dragging its butt as it goes across this surface. Okay? What's it actually migrating to? It's migrating towards a chemical agent that is in high concentration, bless you, up here. And so this is a chemotastics experiment where you put a chemical in a strong signal, a macrophage chemotactic agent, and it moves towards it in a petri dish. Okay? What else? This is a slide that I just added. I think, isn't it on your pictures or no? Okay. I added it earlier in the semester before most of you looked far enough ahead. This is actually total hip replacement on the background. My kids thought it was the surface of the moon. Like, I didn't know you researched the moon, Dad. It's like, no, that's actually a hip replacement. Like, oh, so there's blood. It's like, well, not in that picture. But typically, yes, hip replacements can be a little gory. So the surface is a cobalt chromium alloy metal. It should be smooth, right? You can see a little bit of pitting right here, but like right in this region, it's nice and smooth. And then you see these giant pits. These giant pits are actually macrophages from a total hip implant. And these macrophages have taken residence on this cobalt chromium alloy metal, and they're digesting the surface away slowly. This is on the cover of a biomaterials journal. This came from Syracuse University, and I was reading the article, and I thought, oh man, this is perfect. I'm just going to throw it in there. Furthermore, look at what it says down here. These cells generate an oxidative burst-like reaction, generating reactive oxygen species using Fenton reactions that can directly corrode this cobalt chromium metal alloy in the body. That's what we just said. I just didn't show you a hip implant, but it does it with everything. If it can digest metal, want to bet it can kill bacteria? Pretty cool picture, huh? So the last slide for the break that I'm going to leave you with is this. I wanted to show you relative time here, activity level relative here. You can see time course. The first thing that shows up in inflammation is edema. You see that in the first couple of hours. And then this comes on and fades, followed by the second peak of activity, which we said was the infiltration of neutrophils being recruited into the tissue. Okay, and then we appropriately characterize that the next thing that happens, shown by this graph, is the recruitment of monocytes in circulation, or tissue macrophages, if they show up in the tissue. And here's the tipping point. If this curve comes down and fades, then your inflammatory response is acute, and you've healed the tissue. If this one stays up and remains elevated and continues out, now you have chronic inflammation. We'll talk more about that in the second part of the lecture. Okay. Let's take a break.